Okay, ladies and gentlemen, good afternoon. My name is Mukhtar Aliyu. So today we are going to look at the aspect of bi-reaction mechanisms in advanced enzymology. So without wasting much of our time, if you look at it from the screen, it is bi-substrate reaction. So from the word bi-substrate, if we said bi-substrate, it means that we are using two substrates to catalyze the rate of the reaction. So look at it. Bi-substrate reaction is when an enzyme is catalyzing a reaction involving two substrates and yield two products. So it is called as bi-substrate reaction. So what this means is we have different types of reactions. A reaction can be carried out by one substrate, one product, two substrate, three product, three substrate, two product, three substrate, one product. But in this case, we are talking specifically to bi-substrate reaction. It means that it's when an enzyme catalyzes substrate A and B to produce P and Q. So that is what it means. So you have substrate A plus substrate B, so to give you substrate P and Q. It means that you are using two substrates to produce two products using a single enzymes. So ladies and gentlemen, most of the reactions that take place in the human body are bi-substrate reactions. Like for example, if you look at the most of the glycolytic pathway, you also use bi-substrate reaction. Like for example, if you can take glucose to produce glucose 6-phosphate, you are using two substrates and producing two products, because in the reaction you are using glucose plus ATP to produce glucose 6-phosphate plus ADP in the presence of enzymes called hexokinase or specifically glucokinase. So that is typical example of bi-substrate reaction. So when you are talking about bi-substrate reactions, it means that you are using two reactant to produce two products using a single enzymes. And as I said, most of the metabolic pathways, if you look at the metabolic map of humans, you will realize that most of the reaction they are bi-substrate reaction. That is why you can see clearly in the slides approximately 60% of the reactions account for biochemical reactions. I hope you understand. So bi-substrate, it means two substrate then two products. That is the meaning. Like for example, in this case, it is either transferase reaction in which the enzymes catalyze the transfer of a specific functional group from one of the substrates to another. Like for example, look at this one, PX. If you look at it, you take or you transfer the group from the P to B. So that is what exactly happened. So transferase enzymes, mostly from the classification of enzymes, we have transferases. So when we say transferases, it means that a group of enzymes that transfer a specific group from one substrate to another to produce a specific product. Like for example, in a situation where you have glucose plus ATP, so you are transferring a phosphate group from the ATP and loaded it to glucose. That is why you have glucose 6-phosphate and ADP. So most of the transferase reactions involve the bi-substrate reaction mechanism. So bi-substrate reaction is a oxidation reduction reaction in which reducing equivalents are transferred between the two substrates. So ladies and gentlemen, From here you can see that most of the bi-substrate reactions are redox reaction. When you said redox reaction, it means it's a reaction in which both oxidation and reduction take place simultaneously. So most of the bi-substrate reactions are redox reaction, so in which a reductant equivalent transferred between the two substrates to produce your specific product of interest. That is for this. Then example is look at this one: hydrolysis of peptide bond to bi-trypsin in the transfer of the peptide carbonyl group from the peptide nitrogen atom to water. So look at it. This is a polypeptide change, and this is a water, and this is your trypsin. So you know that trypsin is an enzyme that involved in the hydrolysis of proteins. So you see, when we say hydrolysis, it means that you are using water to produce a specific product. So you see now, if you can now look at this one, you transfer the peptide carbonyl group from the peptide nitrogen atoms to water. So you see this group, the carbonyl group, this one is completely transferred to water. So you see we have this one. So you see, if you look at this one, you understand that we have two substrates. We have this polypeptide and we have water. And if you now look at this, this is carboxylic functional group with amines. So you see, you use two substrates to produce two products. So it involves transfer. So it's transferase enzymes, which is trypsin. Okay. Then the next one, is so Cleland is a scientist that introduced the nomenclature system for representing enzymatic reaction in bi or in multi-steps reaction. Like for example, generally in multi-reactions, the substrate is normally represented by letter A, B, C, and D. So it means in a situation where you have four substrates, so you are going to represent them using letter A, B, C, and D. And the products is represented by letter P, Q, R, and S. So the stable enzymes forms are designated. The enzymes used in the reactions may be designated using letter E, F, and G. So the E is your initial enzymes, while F is the intermediate enzymes, and G is also represent the intermediate enzymes, because generally speaking, in this multi-steps reaction, most especially like in ping-pong reaction mechanism, when E react with A, so part of the A will still be gone with the enzymes. So that is why it is not longer be considered as a few enzymes, but letter F. Why is letter F? Because some part of the substrate is held together with the initial enzymes, so that is why it changed to F. It means that F contain some part of the substrate with your enzymes E, so that is why it changed to F. Unlike also in G, that is also what happened. Then the number of reactant and products in a given reaction are specified in order. So like bi, uni means 1, bi 2, ter 3, and quat 4. So the reactions that require two substrates and yield two products is called bi reactions. So that is what we are going to look for the purpose of this class. Then types of bi reaction. So we have types of them. So there are of two major types. We have sequential reactions. So the sequential reactions, it means that all the substrates involved are bound to the enzymes before catalysis of the reaction takes place to release the products. So it means that in this case, it's like all the raw materials that are required for the reaction to take place must come together. You have your enzymes with the two substrates, so they must bind together before the reactions begin and the release of the products. So like for example, these reactions of sequential, we call it single displacement reactions. Why do you call it single displacement reaction? It means that all the two substrates must bind to the enzymes and displaced at the same time. So they are binding together, the substrate, and the releasing and the products are also released at the same time. So that is why it's called single displacement. A and B join to the enzymes, then you released the products also at the same time. So that is why it's called single displacement reactions. But in a situation where you are A will bind first, then to catalyze, maybe you are A, then you release the product, then B bind also, then you release it. So in that case, actually, if your product are not releasing at the same time, maybe separately, so we call it double displacement reactions. So that sequential reactions is a single displacement reaction. So as I said earlier, both the substrate and the enzymes forms a ternary complex. Why is it a ternary? So ter means tree. So it means that in this case you have two of your substrate and one of your enzyme. So if you match them, you have three. So that is why they are called ternary complex. So they form a ternary complex. Then it is subclassified into two types. The sequential reactions is divided into, or is also further classified into two. We have ordered sequential reactions and we have random sequential reactions. So in ordered sequential reaction, it means that A must bind to your enzyme first, then before B. But in random, it means that both the substrate A and B have equal chance of binding to your enzymes. So that is why we call it random. So but for ordered, the first substrate must bind, followed by the second substrate. But for the random, both the substrate have equal chance. Both the substrate has equal chance of binding to the enzymes. So you see, this is the ordered reaction. So you see, these are the substrate A and B. So A was bind to your enzyme. So you have enzyme substrate A complex, then followed by B. So you see, A, you have your enzymes A, B, E, A, B. So you see, A must bind first, then B will also come. Then the product will also be released in ordered form. In order, you see, P release first, then followed by Q, then you have your free enzyme. So this is what actually happened. So in order, A must bind to your enzyme first, then followed by B, then you have enzymes with your two substrates coming off together. So this is what they call ternary complex. Ternary, two of your substrate and one of your enzymes. Then you also have your free enzyme plus products phi and Q, which phi release first, then followed by Q. So that's the meaning of ordered. The substrates are coming in order A first, then followed by B, and the release of products are also followed in order. So P release first, then followed by Q. Then examples, simple examples of this. Look at it. Enzymes. Lactate dehydrogenase enzymes, which catalyzes the metabolism of glucose. You see, in the metabolism of glucose, you see pyruvate converted to lactate. If you now look at this one, you understand that you have two substrates. You have pyruvic acid, then plus NADH. So you see you have two substrates, and at the same time you have two products, which is lactic acid plus the NAD plus. So you see you have two substrates and you have two products, that is bi-bi. So because it is ordered, the first substrate must come first. You see, the first substrate in this reaction is NADH, so it will bind to the enzyme first, then followed by the pyruvate, which is the second substrate. Then you have the ternary complex. enzymes NADH pyruvate ternary complex. Then you will have your ternary products, which is enzymes, lactate, and NAD plus. So the lactate will release first, then followed by NAD plus. So that is a ternary complex. Then we have random sequential reaction. So in random, you can see that you have A here, you have B, then you have B and C. So what does that mean? It means that A and B have equal chance of binding to your enzyme. So A may come first, and at the same time B may decide to come first. Then maybe, so that is just the meaning. The substrate have equal chance of being bind to your enzymes, and also at the same time the products have equal chance of releasing. So you see, phi may release or Q at the same time, or Q may decide to release, then followed by phi. So that is how it is. Then you generate your free enzymes. And then you see, this is a typical example: creatinine kinase enzymes, which catalyzes the substrate creatine and ATP to form the product phosphocreatine and ADP. So you see, it's a typical example of random. So ATP or your creatinine has equal chance of binding to your enzymes. As you can see, the enzymes may decide to take out the ATP, or the enzymes can decide to take out the creatinine before you have your ternary products. You see, and at the same time, the product also has the equal chance of being released. You see, it may decide that ADP will first release, or the creatinine phosphocreatine. So that is it. Then the next one is ping-pong reaction mechanism. So in ping-pong mechanisms, is one in which one or more products are released before all substrate have been added, and known as ping-pong reaction. So it means that if the first product bind like A, so it must be catalyzed first to product before the next substrate bind to it. So that is why you have A and B substrate. So if A binds to your enzyme, so it must be catalyzed the initial substrate that binds into product before the second substrate binds to it. And then the next thing is characterized by the change of the enzymes into an intermediate form when the pair substrate to product reaction occurs. So as I said, actually I think I clearly make my explanation here that in ping-pong mechanism, the first substrate that binds to produce the product, it's left some part of the substrate with the enzymes. That is why it is intermediate. So that is why it changed from letter E to F, or from F to G. Okay, then that is ping-pong. So this is a typical example of the ping-pong. You see A bind to E, so A substrate A bind to your enzymes, then you release your enzymes. So you have your enzyme substrate A complex, then the A will now convert your substrate to the P, which is your product. So you see, the product must first be released, and you now have your enzyme with some element of A with your enzyme. So that is why it changed to E asterisk. Do you understand? And the E asterisk will now bind to the product B, and the product B will now take up the component of the substrate A in enzymes A and to release the product Q. Then you now regenerate your E again. Okay, and then this is a typical example. See, you have aspartate plus alpha ketoglutarate. So in this case, like now look at it. Okay, you have your enzymes. So the aspartate will now first bind to it, then to produce enzymes amino group oxaloacetate complex, then the oxaloacetate will now be released. So you see some part of the aspartate, which is the amino group, is now added to the enzymes. So that is why you will not regenerate your E. That is why in the previous slide we have E asterisk. So the asterisk, as I said, is indicated that some part of the substrate still remained with the enzymes. So this element that held with the enzymes will now be what? It will now be used as a donor to the next substrate. You see the oxaloacetate will now react with the intermediate enzymes to produce the glutamates. So this is it for ping-pong. So this is the reference. I hope, ladies and gentlemen, you like this video. Please read very well. Thank you. Happy reading.